I'm Christine Loki from PXE International, and I am delighted tonight to have Patrick Terry with us to present the 2012 PXE Research Update. Patrick, of course, is a co-founder of PXE International. He's a volunteer who has done everything in this organization, and he's also chairman of the board of PXE International. He is the father of 2 young adults with PXE. One of them, Elizabeth, is our current executive director. He has worked on basic laboratory research and clinical translation studies for PXE, and he also works to support all the research activities of PXE International. Professionally, Patrick works in the biotechnology industry, and he's joining us from his office right now. Pat, welcome.
Hi, Chris. Thanks, thanks Terry and Chris for putting this together and for hosting this. And my objective today is to do a similar update that we did this time last year. So welcome everybody, and thank you for giving me your time and attention. So what I'd like to do is present off some slides and some talking points and cover if you want to switch to the next slide, Terry, slide 2. So I'll highlight some recent meetings and some workshops and some learnings coming out of these recent events, and then provide a research update both on discoveries, activities, and priorities, and have some time at the end for some open questions and discussion. Or during this presentation, if you want to jot down your questions along the way, I'll try my best to answer those. But maybe just to add on a little bit from Chris's introduction, I am not a trained scientist or physician. I like to say that the initials after my name are JAD, which stands for just a dad. But I've been involved now with basic research and genetics and clinical research because of the diagnosis of my 2 children a long time ago, no longer children, but young adults, and been involved with spurring on research, directing some research, being kind of a citizen scientist now for over 20 years. So I've been involved with some applied aspects of this research, as well as been involved with helping these researchers with research priorities, hypothesis, some of the lab work itself. I've since now gone on to join the biotechnology industry and do this professionally, even though I'm not formally trained. But this is all because of PXE has influenced my career path and my life choices, and so it's both exciting as well as stimulating about the science and where we're progressing and what this means for PXE. So what I'll highlight here is what PXE International, the organization, is focusing on, where we are contributing our money and our resources internationally with researchers of all types. So with that preamble, Terry, you can go to slide 3. And the most recent event, so just last week, there was this biannual meeting called the ABC Binding Cassette Research Meeting, and this takes place every other year, and it's usually held in Austria. And this is a scientific gathering of a week-long program which approximately 300 scientists from around the world and researchers come to talk about this field of what's called ABC binding cassette, and I'll talk a little bit more about some of this terminology. So it doesn't make a lot of sense what ABC stands for, but I'll explain what that means and why we're doing research on multi-drug resistant proteins, and why that matters to PXE, and put this into some context. But this is a major meeting in which this is the 4th year in which we've supported this meeting and participated in this meeting. It's in a beautiful spot, so that's certainly nice. And spring had arrived in in the Alps and in Austria, so it was beautiful weather. But ultimately, this is a research meeting with highly enthusiastic participants that, when they're not in the research meeting themselves, they're talking about research. When they're outside the meeting and they're up late at night talking about research, it's a pretty intense week-long activity, because that's all these people talk about is their posters, their work, their research. So it's not fun and games, but it's highly serious, and we really enjoy this meeting for a variety of reasons, but most of which because they make a very strong emphasis on sponsoring students and young researchers. So a lot of our contribution to this meeting goes into support students to attend this meeting. And they have 90 to 100 young researchers participating under sponsorship. So ordinarily, these individuals would never have the resources or the time to attend such a scientific meeting. But it is a wonderful mentoring program, and some really brilliant young individuals that are leaders in their particular field or discipline come and participate in this meeting, and have certainly got highly interested in PXE, and I'll talk more about why that is. So this update will include what took place at this meeting and some of the takeaway activities that have emerged from this ABC 2012 research conference. So slide 4. This is just to highlight that when we started supporting this meeting, we were one of 3 contributing groups. So the ultimate underwriter for this research program was FASEBS, and it's the Federation of European Biochemical Societies that underwrote the majority of this meeting, because certainly it's not inexpensive to bring 300 scientists to a conference center for a week. So, but now here in the 4th year, there are multiple other supporters of this meeting beyond PXE International, including pharmaceutical industry and other major institutions. So it's a growing meeting and a growing group of involved groups, because of the caliber of the research and the quality of what takes place at this meeting is being recognized broadly. So the slide 5, please, Terry. So I want to highlight also near the end of last year, there was a PXE workshop that we sponsored in Budapest, Hungary, in collaboration with the Institute of Enzymology at the Hungarian Academy of Sciences, that had a specialized PXE research and clinical workshop, and there were 40 scientists that gathered from around the world for that 2-day meeting, or the day-and-a-half meeting in Hungary. So I'll be talking a little bit about what happened at that meeting, because this is the first time we've had an opportunity to talk about the Hungarian research and progress to date. So slide 6. That was the 2 major events, and now I want to talk about the research update itself and what we've done as a group, PXE International, and what some elements of our progress. So on the next slide, when we describe support for research, it's not only financial contributions that we're making to research groups, we're also supplying technical equipment or computers or bioinformatics support, or actually placing staffing or consumables or research tools within these groups. Sometimes research groups are highly funded, so they have research monies, but because of other constraints on how they can spend their budgets or what they're allowed to purchase as an institution or as a group can be challenging. So we can come in and offer them solutions or provide some capital equipment that they could not ordinarily have support for in their current budget. So when we describe research support for the foundation, we do it in a variety of ways and in creative fashion. And in some cases, there are high caliber research groups that we want them to do PXE research work, but for one reason or another, they don't have the time or the energy or the capacity to do that research work, so we can actually sponsor staff and put staff in their laboratories and in their research groups, so PXE work can take place when they didn't have the time or the personnel to do that research. So that's just, I wanted to highlight that there's a variety of ways that the Foundation thinks about support. The next slide, please, slide 8. Here's just an example of some, often in scientific enterprises, what is a badge of honor is certainly to get publications and have what's called peer-reviewed publications. So there's a manuscript and a finding, a paper that is written and submitted to a journal, and then it's reviewed by independent experts in that discipline, and edits are made, and then the research is published. So the broader community can have access and learn from scientific progress or clinical progress that's made, or certain hypotheses that are advanced, and this is called, producing literature is fundamentally how knowledge is shared in the scientific community. And in mid-2011 and 2012, there's been multiple publications around PXE, both the clinical research, the biochemical research, the genetics, and some of the animal studies have been published. On the next slide. So the Foundation also collaborates and supports, financially supports research groups around the world. And one of the new research collaborations that we've established has been with the Laboratory of Biochemistry at the University of Amsterdam in the Netherlands. And this is a group of researchers that are doing animal research and biochemical research. There's a pharmacologist who's studying the therapeutic aspects or opportunities associated with alleviating some of the symptoms of PXE. There's basic scientists and geneticists as well as clinical researchers. And this is an example of a highly prestigious university and investigator that has a tremendous reputation in the field, that had not in the past been interested in working in PXE, but because of this ABC transporter meeting that takes place in Austria each year, 2 years ago we got this researcher interested in PXE research, and now he's committed his entire research group and all of his staff to doing PXE research, and that's quite exciting because he's a highly brilliant man with a lot of tremendous discoveries in his past. So we're really excited to be working with that group. So that's an example of an active research program, which I'll talk more about exactly what they're doing near the end of the slide presentation. The next slide, please. As I mentioned, we are also supporting over multiple years now the researchers in the Institute of Enzymology in Budapest. These scientists have participated in PXE research for many years, have participated in the scientific and research meetings as well as the patient meetings. So you may have met some of the Hungarian scientists if you have attended any of the PXE patient meetings. And this group has been highly committed over multiple years to advancing some of the basic and fundamental knowledge about what's underlying the cause and aspects of PXE, and they're doing animal research and biochemical research and basic research as well. So they've expanded their group, they've expanded their collaborations internationally, and almost, not quite, but almost entirely this research group has committed themselves to PXE research currently. So that's also an exciting development because this is a high caliber group that's highly respected internationally and are doing some really terrific work for us. The next slide, please. Then finally, the other major research collaboration and sponsorship that we're working with is the Institute of Molecular Medicine at Jefferson Medical College in Philadelphia. And that research group has made some phenomenal discoveries and has led the way on some of the research discoveries and advancements, which I'll talk about later in the presentation, but they also have multiple disciplines of scientists and clinical researchers working on PXE. So based on this description and the staff activity in all three of these groups, it appears like these research groups are doing the same thing, right? Because the same disciplines and the same types of scientists are working on PXE. And to some extent, that intuition or that first glance is true. We do have some overlap. We do have some competition. We are supporting some duplicative work, but these research groups are coming at it from different disciplines, a different sense of urgency, or have a different perspective or hypothesis that they're trying to advance. So rather than placing our bet on one group or one set of researchers, we're supporting multiple opinions and multiple research activities because we want to accelerate this PXE research as quickly as possible and as efficiently as possible. So while we help these groups to collaborate and work with one another and exchange ideas and exchange students and staff and so forth, we're also recognize that it's important to have a healthy sense of urgency and competition among these groups to keep everyone focused on doing the best science that they can do. The next slide, please. So what the next set of slides that I want to share with you is what we think we know. Ultimately, the challenge in science is that something is thought of, experiments are done, some results are derived, and people make educated guesses on what this information and this data means. Then they publish it in the literature that I referred to, and they have a peer publication, and some other research group reads that discovery and then tries to replicate it, and then they can publish whether they were able to reproduce those same results, or if not, why not. And this is basically how you test ideas and discoveries, and that's why science sometimes advances fairly slowly, is because of this process of publication and testing and retesting. So what I'll share with you is what we think we know. It's certainly not true in every absolute sense of the word, but it is the current opinion held by the researcher and the community generally. Next slide. So this is a very simple slide of 3 bullets of what PXE is. So in the most generic and abstract terminology, this is what's called a systemic genetic disorder. And systemic means it's affecting multiple systems in the body, but ultimately it is because of a genetic alteration in human biology that has caused this disorder that we describe as, or clinicians describe, because of a phenotype that you have pseudoxanthoma elasticum. And PXE was initially described by clinicians, well over 100 years ago. So there's a long literature and many case studies and patient examples of what we think PXE is and how it presents itself, and it's genetic in cause. And it was always believed to be a genetic disease. No one knew until 2000 or in late 1999 really what was the genetic causality of this disease. And ever since those discoveries in 2000, we've been working to try to understand what are the genetic mechanisms, how is the disease brought to fruition in the body, and what does this all mean for patients. And ultimately, what's been described and what's been believed now is that the genetic disease causes a loss of function. So, and I'll describe what a loss of function means in some animations in some later slides. But predominantly, it's a loss of function in the liver and/or the kidney. So it hasn't even been isolated to be just the liver or just the kidney, but most of the scientific community believes it's predominantly a liver disease, that there's a gene or a protein in the liver that is not functioning. And because it's not functioning, all of what we describe as PXE is a result of that loss of function. So predominantly it affects, this loss of function affects what's called the elastic fiber in the body. And these are cellular, subcellular structures that bind cells together, that create the architecture for cells to be in relationship to one another, that allow tissues and other bodily functions to operate along these because of these elastic fibers or these collagen fibers. And elastin and collagen are just basically structures that allow tissues to function in certain ways. And elastin and collagen work really well to be resilient and elastic type of structures that allow stretch and recoil, that allow, if you pull on your skin, to allow the skin to snap back, or allow blood vessels to contract and pump blood through the body. So there's actually there's a form and a function to these fibers, and these fibers get affected in certain tissues in the body that are very crucial to individuals, and certainly crucial to how our skin functions and how our eyes function and how our arteries or our gastrointestinal tract functions. So these are important components that actually end up getting damaged because of this loss of function, and we'll describe that a little further on. But next slide. So PXE genetics. So we've been able to firmly describe in the literature what's called autosomal recessive. So again, this is another complicated word of describing inheritance. That means that you need 2 copies of an altered gene to produce this disease. So both a mother and father would need to contribute to transmission of altered genes and produce this loss of function. And we've been able to prove that it's not a dominant disease through case studies and genetic studies, and being able to firmly describe that this is a recessive disease. Next slide. This is just a complicated picture of the nomenclature or the identification of genetic alterations in the gene. And each of these have a specific location and where they reside on the gene and the protein. So this is literally a map of the thousands or over 1,000 alterations in the PXE gene that have been discovered and described by PXE International in their research collaborations. Next slide. And here's a cartoon of the protein structure, and it's simply a 2-dimensional map of the protein in a simplified cartoon format, and where those genetic alterations alter an amino acid in a protein structure, and we've mapped those. And in these alterations to the protein structure cause different behaviors in this protein. Sometimes it destroys the function of the protein entirely. Sometimes it just inhibits the function. Sometimes it alters how it traffics and gets to the proper place in the cell membrane. There's multiple behavior patterns that are associated with each of these alterations. So we've gone on to find these alterations and to describe them in what are called experimental models or cellular models. So there's about 5 or 6 experimental models that have been used in yeast and in flies and in zebrafish and in mice and in various insect cell lines to be able to discover what these alterations actually do, how they affect the protein, and do they avail themselves to any type of manipulation or therapeutic fix. So it's ultimately, all of this research is focused on certainly knowledge gain, but also very much focused into, are there types of alterations to the proteins that cluster into certain types of families or behaviors that we could actually correct or fix and alleviate or delay the onset of PXE or alleviate some of the symptoms of PXE. Next slide, please. This is slightly out of order, but so pseudoxanthoma elasticum is clinically described as a late onset disorder, and that's not necessarily true in all cases, but in most cases it is because what we describe as PXE or pseudoxanthoma elasticum is what is observed by a clinician or observationally, saying, well, you have skin lesions or ocular lesions, and these alterations to the elastin and collagen fibers in the body are classic PXE. But that has taken decades for that to actually happen. So that's what's meant by a delayed onset disorder, that it takes time for PXE to develop, and the phenotype or the clinical expression of this disease is very highly variable. It can even be highly variable within families. And there's been a significant amount of work around what in the genetic arena about what we thought we could do is be able to find these genetic alterations and describe them as a genotype. So certain patients would have a certain mutation which would equal or equate to a certain type of disease status, or most likely to have this or that complication. But we've been able to show that that's not the case, that you could have two individuals with the exact same genetic alterations or genotype and have significantly different disease. So this linkage between a genotype and a phenotype is highly variable as well, which means then that there are other modifying factors, either other modifying genes that alter how the disease progresses, or significant other influencers on how the gene actually works or doesn't work in the body, or how the environment influences the genetic function or the protein function. So we're also coming out of the meeting that we had here last week. There's new research programs going to be launched in the months ahead about understanding these modifier genes and some experiments to make much more sense out of why are these genotypes and these phenotypes so variable in the patient population. The next slide. And then I'm going to talk a little bit more about the gene and the protein. So the next slide. So ABC stands for ABC transporters, or a class of genes and proteins. And ABC stands for ATP binding cassette. That's where the ABC comes from. And ATP is a chemical within the body that's actually like it's like gasoline. It's a fuel that actually provides energy, kinetic energy, for cells to function. And I'll describe why that's important for PXE. But I just wanted to give you a little background here that these proteins, these ABC transporters, are highly conserved through all of nature and all of living organisms on this planet. It's essential to life that these proteins exist in bacteria and plants and humans, and evolutionary, they've adapted to many different circumstances, and they're highly variable in their function. They cover a whole gamut of functionality within different species, and even within the human body, that the same gene and protein can operate quite differently depending on which tissue or organ it's expressing itself. Next slide. So this is a simple cartoon of the function here. In the ABC protein is a transporter and an active transporter. So you have this ATP that binds to these two pumps, these two gray boxes, these ATP boxes. And when ATP binds there, it causes a chemical reaction, and they operate as if it's a two-piston engine, that these chemicals bind, and then the protein actually works as a pump. And these yellow dots are an example of, so something that's inside the cell is pumped outside the cell, or something that's inside the liver is pumped outside the liver or kidney. And that's what this protein does. It's an active transporter of a certain substrate or a certain set of molecules that are important to healthy function. And in the case of pseudoxanthoma elasticum, this pumping and this protein is not working properly. Because it's not working properly, because of this loss of function, then we have what we are describing as pseudoxanthoma elasticum. Next slide. Here's another cartoon that I drew, so it's pretty crude, of a slice of a cell. So you've got this, on the bottom there you have this big orange nucleus, so that's where the nucleus of the cell and the DNA is housed in the nucleus, and then you have this space or this cytoplasm. This is the inside of the cell, and then you have this purple and green and blue membrane. The ABC protein sits as a channel in the what's called the lipid bilayer. So there's these two layers that make up the cell membrane, and this protein spans this membrane, and there's a molecule inside the cell that gets actively transported outside the cell. So this is just, I want to put it into a little bit of context of how this protein actually works. It's a channel, it's a pump, it's a pass-through that is not functioning in PXE. Next slide. The other complicated, whoops, I just messed up my own computer here. The complicated aspect of this as well is that the family of proteins is a large one. So there's 48 different ABC transporters. So all of these transporters are in the same family. They all have various and differing functions, but it's a quite large family of genes and proteins. And that creates a tremendous amount of opportunities for research, and as well as a tremendous challenge, because what's been discovered and what you'll hear later is that what was thought to be, if you were part of the same family or the same branch in the family, that you would operate the same way. And that turns out not to be the case, that these proteins, while they're similar, do not operate the same. And that's certainly somewhat disappointing for us, because there's a lot of these proteins that have been studied a lot longer than PXE International has been involved, in a lot longer than PXE has been studied. But nonetheless, we've, over the last few years, have been supporting research in 16 of these proteins in hopes of discovering what might be actually taking place with the PXE protein. And then the Hungarian group has certainly been the most instrumental in that research. They've done the majority of the research work across these different proteins, and tried to use studying these various proteins to help inform what's happening with our protein. So this year, we've moved away from sponsoring all of these different proteins and focusing on ABCC6 with a new emphasis on, rather than focusing broadly on all of these different proteins, more narrowly focused on the protein of interest. And there's a variety of reasons for that, because there's new opportunities, because of what I'll describe a little bit later about the animal model. And let's see here. I'm trying to load up the, I ended up kicking my my computer and turning it off, so I have to turn my computer back on and get the screen back up. Terry, I apologize.
I noticed a few people joined us a little bit late, so I can just reiterate that if you have any questions about anything Patrick is telling us, you can type your question into the question pane. It should be found on the right side of your screen in the control panel. Hit send, and we will see it at this end.
So bear with me for just a second
We will
do
do that.
Any questions?
We do have a question. Why are we focusing on ABCC6?
Yes, so ABCC6 is the gene that is directly linked to pseudoxanthoma elasticum. So when the genetic research was taking place in 1999 and 2000, the PXE patients were sharing the same section of chromosomal DNA. So that everything was there was a certain section of DNA that was similar for all PXE patients. And it was narrowed down to 6 genes in that section of DNA that all of these patients shared. And then there were 4 or 5 groups that then tried to identify, well, which one of those 6 genes is linked to pseudoxanthoma elasticum. It was found by 2 groups simultaneously that ABC6, or MRP6 at the time is what it was called, was the gene that was responsible and linked to pseudoxanthoma elasticum. So I'm back up. Terry, could you advance to slide 23? So this is where the research community is today. So this is brand new information. This is the community's consensus about what is the understanding of ABC transporters. So these are understandings / assumptions. Again, with my beginning caveat when I entered this meeting is that none of this is absolutely certain, but this is what the community thinks is true. So this is what they think the understanding is in the context of health and disease and therapy. So we thought that genotype and phenotype would link, that if you had a sequence in a gene, you would have a certain function and a certain disease outcome, and that's proven not to be true. So the answer is no. There's no easy linkage, that biology is really complex and it's not as simple as we would like it to be. Because we studied 16 different proteins at the same time, we assumed that if a protein was structured the same and operated in the same way, that it had this pumping mechanism or this active transport, that they would have the same mechanism, that if you understood one, you would understand the family. And that turns out also not to be true. We also thought that every single membrane transporter would have a specific substrate or a certain chemical that it was optimally designed to work on or to transport. But we've also discovered, quite frustratingly, that no, that these proteins actually, some researchers call them promiscuous, meaning that they'll transport all kinds of things or anything that presents itself it will transport. So when you're trying to find out what's missing or what a particular transport is supposed to be trafficking or shuttling across the membrane, if it shuttles a lot of things, then it doesn't really tell you exactly what out of those bunch of things that it transports are important to PXE or not important to PXE. So there's still a big maybe there. It turns out it's not unlike if you have a rain collector barrel in your yard. You have all of these different outlets on that rain barrel, and there's one at the bottom, and it works well, and it allows you to water your plants. And then there's a big outlet at the top that if the barrel overflows, then that outlet works and allows all that excess water to drain out. And some believe that these transporters operate on the same mechanism, that in a perfect environment, the protein is specifically designed to shuttle a specific molecule or substrate. In other circumstances, if the cell is under stress or there's abundance of a certain molecule inside the cell, that then the proteins and these transporters will help to move that material out of the cell. So that makes it a little bit more complicated to find out exactly what's being transported, but that seems to be universally true for all of these proteins. The other frustrating thing we've discovered is we thought that if you had one ABC transporter, you had one function. And that also probably turns out not to be true. This is a circumstance that evolution has been very adaptive about using the same protein for a different function, depending where it is. If it's in a muscle cell or in a skin cell or in a neuron, you're using this same protein but for a different function, depending on the tissue or the organ in which it's expressing itself. So also we have to be very careful about how and what these proteins are doing in the skin and in the liver and in the muscle cells and so on and so forth. And again, these are highly variable ultimately. So even in model systems, so we've used insect cell lines, mouse cells, various liver cells, zebrafish, flies, yeast, and these proteins all function in those different organisms, but they function quite differently and behave broadly in these different model systems. So these proteins are quite adaptive and quite versatile, which makes it hard to be able to describe exactly what they do. So that's not to be too dour on this. We're making progress. We've made a lot of research discovery and a lot of progress, and even knowing for certain that some of these things, of these 5 truths, are real, are really helpful. Rather than having questions in every direction, we're now getting a little bit more certain around avenues of research activities and opportunities for further discovery and further knowledge gain that allow us to get a little bit closer to understanding exactly what's happening at the biological level and perhaps how we can fix it. The next slide.
Pat, we have a couple questions about that from a few people. Okay. Just to be clear, are you saying that researchers think that ABCC6 is the protein that is causing, whose malfunction is causing PXE?
Yes. Yep.
And someone else has asked, do we know, have any idea what it's transporting or not transporting?
Yeah. So that's the $6 million question. So. We don't know, and I'll explain, the next set of experiments that allow us to get us there. So this slide 24 is then again some other mechanisms and some other challenges that are being worked on by thousands of researchers. The advantage here of having so many different proteins and having these proteins quite similar in function and in structure and in activity is that there are researchers that have no interest at all in pseudoxanthoma elasticum and will never have interest in it, but have interest because these various proteins, when they lose function, cause hundreds of other human diseases. So there's lots of disease researchers and basic scientists working on these proteins for different reasons, and they're quite active. So we can skip that slide and get on to the protein activities that are taking place, and these are fancy models of what the proteins are doing, how they function, which once you understand some of the shape and their structure, you can start making some guesses or some hypotheses about these substrates of what these proteins are capable of transporting. If they're arranged in this manner and function in this way, then physically we can define a limited universe of what these proteins can function as. So there's lots of new experimental models around the structure and function and biochemistry of these proteins that we're supporting and other groups are working on. Next slide. I'm almost at the end here. So here are the research activities. We'll highlight here the next slide. So there's active research taking place in what's called the animal model. So there is a mouse model for pseudoxanthoma elasticum. So these genes have been manipulated in the mouse to produce pseudoxanthoma elasticum in the mouse. And the good thing here is that it's almost identical to the human disease. So this is a very good research model to understand what is taking place, how it's taking place, how this gene and protein functions in the mouse. And if you click the next, you'll have an animation here that we're studying the difference between normal mouse, or what's called a wild-type mouse, and a PXE mouse. And there's been some phenomenal research experiments done by the Jefferson group, where they've taken a PXE mouse that has disease, has calcification of the arteries and macular degeneration and so forth, and has done what's called conjoining, literally sewing together two mouse, a wild-type mouse and a PXE mouse. And when you do that, these mice are sharing circulatory systems and sharing liver function. And the PXE symptoms in the PXE mouse disappear. So basically a normal function of the protein in the liver and the kidney reverse disease in the mouse. So that's kind of proof that this gene is indeed responsible for pseudoxanthoma. When it's working properly, it can eliminate the disease altogether. So we're comparing these two mice, normal and the PXE mice, and now we're entering into a program of comparing non-PXE patients and PXE patients and saying, okay, this is like comparing the Sesame Street game, is what's the same and what's the difference here. And being able to compare what's same and different in mice and what's same and different in humans will allow us to understand exactly what's not working and what this protein is transporting. Next slide. So coming out of the Austrian meeting, we have a research program with the Netherlands group. We have a meeting on May 4th with European patients are gathering, and we're collecting blood and serum from these patients, and we're going to do a matched experiment of looking at mice in a certain way and looking at humans in a certain way, and they have some novel technology and some research approaches that we are hopeful that we'll actually be able this year be able to discover what's being transported or not transported by the MRP gene. Next slide. And this is, I think, my second to last slide. The foundation has also been conducting an epidemiological research program, and epidemiological means we're just tracking patients over time, and we have a survey that individuals and families have participated in and participated over a long period of time. And if you're interested in participating in that research program, there's many ways to do so, and there's a list of them here, and you can go to the website and figure out how to participate. And this is highly valuable because we can understand across a broad spectrum of patients, how does the disease manifest itself, how does it progress, how did it behave in you, in your life, and it allows us to really characterize this disease in a much more robust way than it's ever been done before. Next slide, and then I think that's it for my presentation. Is open for discussion now. That's the update of where we're at. We're really excited about the two research initiatives that came out of these recent meetings. This is the proteomic study of mice and humans, and then the modifier genetic research that's going to be taking place. So those are the two hot off the press research programs that have emerged recently. So I'm open to any questions that anyone might have.
Thank you very much, Pat. We do have a couple questions. One of them, going back to the epidemiological study, I know you're not a physician, but someone has asked about a certain condition and whether it's related to PXE because of loss of elasticity. In this case, aortic aneurysm. Is there any evidence that that is caused by loss of elasticity in the fibers of the aorta?
Yeah, so it's a controversial area because there's certainly when you have a systemic disease, doesn't mean that's the only disease you have. And if you, for instance, if you have a family history of heart disease, then you can have pseudoxanthoma elasticum and then have familial heart disease as well. And it doesn't mean that PXE has caused your heart disease. There are beliefs. There is some research from Holland in a Dutch group that have made the claim that pseudoxanthoma elasticum causes mid-sized artery disease. That work has not been replicated. As I mentioned, when you publish something, other researchers try to replicate to see if it's true. So this linkage to cardiovascular disease and pseudoxanthoma elasticum has not been reproduced in a robust way. And when you look at the various models, animal models, whether the zebrafish or the mouse model, it does not appear that cardiovascular disease is occurring in those animal models. But as I say that, there's also the challenge that mice are not humans. So there's many PXE patients that have cardiovascular disease or have gastrointestinal disease or other complications that may or may not be induced or caused by Pseudoxanthoma elasticum. So you just have to be careful about putting all your ills and all your challenges and linking them to Pseudoxanthoma elasticum. They may or may not be related.
Thank you. There's a couple questions about the mouse conjoining again. Maybe you could go back to the slide of that, because one person asked if the livers are actually joined, or what exactly happens in that experiment.
Yeah, and it's a pretty crude and it appears brutal, but mice are anesthetized. And an incision is made, so a small incision along the side of the mouse, each mouse, and then the mouse are literally sewn together. So they heal as if they're Siamese twins, so they're conjoined. They're joined together, physically sutured together. So when that suture and that wound, that surgical procedure heals, then fundamentally the mice are sharing biological functions. So when the wound heals, their circulatory systems mingle together, and that simple connection to it's as if you take a PXE mouse and did a liver transplant, just corrected the liver function. And what's been amazing is that the calcification in the mouse, so there's highly elastic sheath around the whiskers of the mouse. So each mouse whisker, where it enters the snout of the mouse, has a little pouch that's highly elastic fibered. So mice navigate the world through their whiskers, so they're highly functional, and they have this highly elastic sheath that gets mineralized only a few weeks after they're born. So they get skin mineralization and these elastic fibers mineralized just like PXE does. And when you join them together with a functioning mouse liver or circulatory system, the PXE symptoms go away. So Yoni Wido and his group 2 years ago published that, which confirmed that Pseudoxanthoma elasticum is a metabolic disorder, that most likely it's related to liver and kidney function. And everything that we observe with PXE is just a secondary event, that ultimately something is missing in circulation in PXE patients, that if that is once corrected, or you can find out what's missing and deliver that to patients, then perhaps you could delay or eliminate the symptoms of Pseudoxanthoma elasticum. So it makes it really challenging to figure out what that is, what's not circulating or should be circulating. And once we figure that out, the advantage of having a metabolic disease means that if you can replace it, then that means you can inject it into the bloodstream or into the patient and potentially have an effective therapeutic solution for PXE. But we're a long way away from that. That's only conjecture on my part, but that's what this mouse experiment seems to indicate.
Sorry, I was on mute. Another related question there has been posed by somebody. What about a liver transplant in a human? Would that correct PXE in a human?
It could. That's the hypothesis. But obviously, getting a liver transplant and going on immunosuppressants could actually be worse than Pseudoxanthoma elasticum. So it's not an easy or an appealing treatment or solution. So there's certainly, we have our ears and eyes peeled for any circumstance in which a PXE patient would have or need a liver transplant for some reason. There's a case report in the literature where you had a healthy individual appear to receive a liver organ donation from someone who had Pseudoxanthoma elasticum, and then that healthy individual ended up developing PXE-like symptoms. Again, that was a highly controversial report in the literature, but that again indicates that there's another piece of data that indicates that the liver is a significant factor in pseudoxanthoma elasticum, and one of the research groups is attempting to do microsurgery in mice to see if indeed they could just take a mouse and do surgical removal of the liver and transplant healthy livers into the mouse and see if that works. But that's a very complicated and difficult surgery to do. So it remains to be seen whether there's going to be more data coming out around just replacing the liver function. Is that all there is, or is there some other factor here? What I didn't highlight is, while we say that the liver is predominantly involved, one of the aspects that's still highly controversial in PXE research community is it's believed that's not the whole story. While it may be a major and significant contributor to the disease manifestation, people believe that you cannot eliminate the importance of ABCC6 function elsewhere in the body. So even though it's not working in the liver, it means it's also not working in the skin, it's not working in the eyes, it's not working in other organs. So just to fix the function in the liver may not alleviate all the signs and symptoms and difficulty with PXE. So that's an open question that still certainly needs to be resolved. But I just want to make that highlight that just fixing it in the liver may be a nice solution for pseudoxanthoma elasticum, but it also may be much more complicated than that.
And it was our medical director, Dr. Berkovich, who did publish that paper about the liver transplant and the acquired PXE, right?
Yes. And that's not, it's not a slam dunk because it's all retrospective, and it's not a really we define the patient and the donor and confirm mutation and eliminate all other potential causality. So it's a very interesting story. It's an interesting case study, but again, it's an N of 1 that hasn't been replicated, and but it's an important piece of evidence, and it was important for Lionel to publish that and to the community to come to consensus that most likely this is a metabolic disorder.
Thank you. Do you know if we have any sense of environmental or genetic factors that might be contributing to the differences of phenotypes seen in the same family? For example, have we studied any twins and looked at what might be making the PXE different?
Yeah, that's a great question, and that's still an unresolved question. And coming out of the meeting last week, that was an important question. So the PXE International and research collaborators are now looking at, now that we know ABCC6 is involved, the genome is going to be reviewed again and looking to see if there's other genes involved and other contributing factors genetically as a first question, because that experiment can be done reasonably quickly and robustly. So we'll find out what possibly other genes are involved with pseudoxanthoma elasticum, and that could give us a better picture on what environmental factors may influence those genes, if indeed other genes are involved or other control mechanisms. So the challenge is the more and more we learn about genetics, the more and more we realize that it's a highly complicated molecule that operates. Just because you have a certain alteration in DNA does not mean you manifest a disease or a sign or a symptom or a trait. That there's other control mechanisms within the genome or within our cellular makeup that can compensate for these different alterations in DNA. So we'll need to do a little work there to discover what other genes may be involved, because then we can start asking and answering the question, well, what other environmental factors are involved? And it's believed that hormones may be a significant contributor to disease and disease severity. certainly other environmental insults like smoking, obesity, lipid layer levels, hypertension, other aspects of normal biology or other disease circumstances may play an influence as well. So we'll be able to make better sense out of the other question about cardiovascular disease, and is it linked, and if so, how is it linked, or is general cardiovascular disease caused by other alterations in other genes, and you just have to you have both. You have genetic alterations that cause heart disease and genetic alterations that cause PXE, because statistically, the researchers believe now that each of us carry 40 to 60 genetic alterations that predispose us to certain diseases. So we're not normal. We are all quite dissimilar. So we all have different alterations that make us unique and individual and make diseases unique and individual. So it can be a big and long and complicated question to be able to explain cause and effect and why disease in a 20-year-old in a family and a disease in a 25-year-old in the family will be quite different, even though they've had the same lived experience and eaten relatively the same things and been exposed to the same environment. But because of their genetic background and because of these other potential alterations, they have disease in quite different manner.
There have been a couple other conditions or genetic conditions described that have manifestations similar to PXE. Are they helping us understand PXE at all? And can you talk about some of them?
Yes. So it's been in the literature. There's numbers of diseases. There's actually PXE research that actually has shown that you've got an individual that's been diagnosed with PXE, but technically, as we currently think about it, they don't have PXE because they've got a genetic alteration in a different gene, and they're only a carrier. They have one good copy and one altered copy of ABCC6, but because they have this combination, this genetic combination, they manifest PXE-like symptoms. You have other diseases of the Mediterranean called beta thalassemia, which is a disease quite different from pseudoxanthoma elasticum. But because of what that disease does to the cells in an individual, it produces PXE-like symptoms. So even though while it's a very different disease, the genetic cause is very different, but its impact on a patient's cells are just like the impact of pseudoxanthoma elasticum. So again, you can observe an individual patient and look at them and say, oh, you have classic PXE, but they could have beta thalassemia and not PXE. So it does help us to understand the biology, and it helps us understand the phenotype or the expression or trait of a disease. Because if beta thalassemia cells behave the same way that pseudoxanthoma elasticum cells behave, then there's a reason for that. And there's an explanation. And that's because these cells become hypoxic. They become very stressed. The cellular environment becomes relatively hostile to normal biology function, and that impacts the membrane transporters in beta thalassemia, and fundamentally there's a loss of function in these transporters that produce the same effect as pseudoxanthoma elasticum. So it's a bit of an overlap, even though these diseases are different, but they're behaving the same way in human biology, and they certainly help researchers understand the total linkage of going from a genetic alteration to a protein function to cellular behavior to how the human disease affects individuals.
Thanks. Another couple questions kind of relating to the mice. You mentioned that mice develop PXE after their birth. So does that tell us anything about the substrate possibly crossing the placenta from the mother to the baby?
That's an excellent question, but the disease, it happens after a few weeks. So the mice, when they're born, do not exhibit PXE symptoms, but The other process here is the age of mice happens quite rapidly. So, mice at 36 or 42 weeks are, old geriatric mice. So in that time window, relative similarly to human biology, so anywhere from the second or fourth decade of human life, you can be diagnosed routinely with pseudoxanthoma elasticum. And that same similar window in the mouse lifespan, diagnosis of PXE can happen as well. So when I made reference to the mouse model being quite similar in the signs and symptoms, is that the timing is relatively the same when mineralization and calcification starts to happen, when skin alterations take place, and when it can be diagnosed with similar techniques that human disease is diagnosed. That's what I meant, is that it's quite similar. These mice are engineered to have no function. So the parents give birth to no function to mice offspring. So these are fully non-functional PXE mice, and there's ongoing work now. Can you take these affected mice and breed them with unaffected mice, and if so, what does disease stop? Do mice that are only carriers, meaning they have one good gene and one altered gene, do they do or don't they exhibit pseudoxanthoma elasticum? Those experiments are still ongoing. And
another question relating to a bit to what we found out in mice. It was an experiment in mice that showed that magnesium had a positive effect and eliminated the development of PXE if the babies were fed a diet high in magnesium. Can you tell us the status of the magnesium intervention studies in humans, and are there any more studies in animals? And is there a difference between the forms of magnesium? Are they giving different forms? For example, magnesium oxide versus magnesium citrate.
Right. Yeah, that's a great question. And yes, again, that type of experiment is attempting to be replicated again. And there was lots of challenges with some of those studies, because it turned out that in certain circumstances the mice were eating their bedding in their cages. So even when you were trying to control the diet and intake of magnesium for the mice, there were other complications. So there are multiple groups working with different mice models to reproduce those findings. And there is certainly the clinical program that is still being organized to do this in a controlled manner with patients to see if a similar finding can be found. So it's still work in progress. And while we support that type of activity, and certainly being able to alleviate some of the signs and symptoms with a do-no-harm approach, with a potentially safe vitamin or nutraceutical or something, that's certainly exciting for us. It's only really dealing with the symptoms, and we certainly just want to keep focused and plowing ahead and actually finding out what the cause is, and if we can truly resolve pseudoxanthoma elasticum. It's not the objective of the foundation to go on and on. We really just want to find a solution here and tame the disease with an effective approach and allow people to not worry about pseudoxanthoma elasticum and live their lives. So if magnesium is part of that equation, we'll certainly support that research as it goes forward. We'll be involved with the clinical study. Once that's formally announced and launched, the community will hear more about that.
And that trial is visible on clinicaltrials.gov. It is not yet recruiting, and they are using magnesium oxide. In that vein, can you tell us what the research objectives of PXE International might be over the next 1 to 3 years?
Yeah, it's we're certainly frustrated. I won't characterize it any other way. We thought that once we had the gene and the mouse model, and being able to do all of these experimental models, we would understand what the substrate is nearly 2 years ago. And here we are 2 years along, and I just described all of the challenges that we've run into. But with this research group in the Netherlands and this mouse human proteomic research project, it's certainly our hope that we find out exactly what the substrate is this year or next. So our short-term objective is to find out what the substrate is. And the purpose there is once we can find out what the compound is or the molecule is, we'll certainly want to find out, can a synthetic version be manufactured? Can a pharmaceutical-grade product be developed? And can a product in a solution for pseudoxanthoma be created? We also want to understand what other factors influence the disease, because if we can identify some of those mitigating factors, genetic factors or environmental factors, we can help patients delay the disease onset or reduce the severity of the disease complications. So basically we want to work to alleviate the burden of disease and to manage this disease as effectively and as quickly as possible. Certainly we got involved with this because of our 2 children, but now our family is much broader than that, and it's thousands of individuals around the world and individuals yet to be born that we're working for, and certainly the next 2 to 3 years is to find the substrate, see if it avails itself to an intervention, and if so, what would that study need to look like, what type of funding, what type of collaborators, what kind of pharmaceutical partners would we need to make that happen as quickly as possible.
Thank you, Pat. We still have a few more questions, but I want to be cognizant of your time. Do you have time to answer a few more?
Sure. If people have more questions and want to stay on, I certainly have the time.
Okay, thank you. You talked about liver or kidney, and but in one slide, but then you didn't mention kidney much after that. Why are we still considering the kidney as a potential player in PXE?
Yeah, so there's been, as we've reported, there's been 2 years ago there was a hypothesis that the kidney would be highly involved, that the kidney worked in vitamin K function and metabolism, and it was believed because that vitamin K had something to do with pseudoxanthoma elasticum and the kidney was highly involved. That those research programs proved not to bear fruit and produced a lot of confounding data and results. So there are some researchers in the community that still believe that the kidney is involved somehow, that it's not, again, it's not just the liver. You have other researchers that believe it's not just the liver and not just the kidney, that it is yes to all of those things, that local gene function matters as well as liver and kidney function. So the challenge is it involved, yes or no, is a difficult question to answer. So rather than try to eliminate, we're trying to ask and answer the question, what's more important? Loss of function in the liver seems to be the most influential and the most linked to the disease course. So we're working hard on understanding and characterizing the liver function. And once we can do that, then I think we can ask and answer the follow-on questions of, well, is the kidney also important, and if so, how much, and in what form is the kidney function important? So we haven't been able to eliminate that liver and kidney and other local tissue function is or is not a crucial component of PXE.
And another kind of general question about stem cell research and developing new retinas. Would any of that apply to PXE in the future?
It could. I mean, generally scientific progress we're all benefiting from. Even the research tools that are becoming available, the types and complexities of the experiments that we've been able to support, all of that has been benefited by progress more broadly. It was thought that any genetic disease would need gene therapy or stem cell replacement therapy, and all of that research is progressing, but it's going to be taking literally decades for that research to bear fruit in a meaningful and impactful way. And that's why we're certainly not happy, but we are taking advantage of the circumstance that if indeed PXE is a metabolic disease, there are tremendous precedents for effective therapeutics being developed for metabolic disorders and for patients that have at birth have a metabolic disease and receive a therapy and have completely corrected function and live normal, happy lives. It certainly lends itself to an approach that is much more here in the near term to have a solution than gene replacement therapy or stem cell therapy. But you know, the eye research is making tremendous progress. The challenge with pseudoxanthoma elasticum is it's not necessarily the retinal cells that are the problem. It's this Brooks membrane, that you have structural defects in the back of the eye that allow perfusion of blood vessels and hemorrhaging in the eye. So even if you were able to replace or continually replace retinal cells, you're just not really dealing with the root cause of the problem. So stem cells could possibly play a role in the future, but it's a long-term research program and slow progress that will, I believe, will have minimal short-term impact on pseudoxanthoma elasticum. So that's why we're trying to, rather than focusing in on all the possible research that could help patients and individuals, we're trying to focus in on what is likely to have the most impact in the shortest amount of time.
Thank you, Pat. And then one final question from a dad, and this kind of speaks to our epidemiological survey and the information we're trying to gather there. Do we know what ages that the more severe effects of PXE generally manifest?
We have data on the various courses of the disease, and we've eliminated what was described as extreme phenotype, because literally what was in the medical literature and the clinical literature was the extreme cases of, young children having cardiovascular disease or having severe complications, and that being described as what's going to happen with individual patients. And that was certainly scary when our kids were diagnosed in the early 90s. We thought they were going to be going blind and have cardiovascular disease and not live past 30 years old. And that turned out obviously not to be the case. And it's not the ordinary experience of PXE and PXE patients. So the epidemiological study is important to be able to describe these scenarios and to make sense out of individual cases. That because an individual has PXE, diabetes, is a train smoker, and obese, probably has more to do with the severity of their PXE manifestations than just having PXE. So the epidemiological study is important to be able to describe all these other cofactors of disease and disease severity. So having families involved is helpful, because if you come from a family that has 4 generations of myocardial infarction or other disease conditions, then that's important to know, because again, you can have various diseases at the same time that you have PXE. So some of your complications or some of your clinical and disease challenges might be other diseases that are much more important and much more complex than PXE, and we can help make sense out of some of that with the epidemiological studies. Have families participating is helpful to get medical backgrounds and medical histories on the mother and the father, and getting family histories and capturing this type of data is important for everyone to truly understand and be able to answer that question with confidence in the future.
Well, thank you. With that, I think we've concluded our webinar, and I'd like to talk about a few upcoming things. But first of all, thank you very much for taking the time with us today, Pat. We've really it's really been an informative webinar. We have a couple things coming up. We have a health challenge currently going on, and one lucky person will be winning $1,000. You can find details about the health challenge on Facebook or on our website at that URL right there. And we have biennial member conference, which will be taking place September 22nd and 23rd this year. That's a Saturday and Sunday in the Washington, D.C. area, just the suburbs in Maryland. And we hope many of you can join us there.